Hi, year 11s and 12s. This is our third PowerPoint in the DNA and proteins topic. This week we're going to be looking at enzymes. So last week we looked about how protein synthesis occurred, so how to make proteins. Enzymes are a type of proteins that are really important biologically, and we need them in our body a lot. So this week we're focusing purely just on enzymes. There will only be one video this week. It's not broken up into multiple videos, so just one PowerPoint, one video. Let's begin. As per usual, here's your Saifs Outline need to know document. So you can see there's 4 main points we're covering here, with each with a little bit of detail of how much you need to know below. As per usual, I'm not going to pause the video here. I'm not going to stop the video here. If you'd like to note these down, you'll need to pause the video and do that in your own time. But let's start the actual PowerPoint. So what is an enzyme? First off, we have to look at what a cell is, because we haven't covered cells yet as a topic. So a cell is like a chemical factory. There's many different chemical reactions taking place in your cell at all times, and without enzymes, those reactions wouldn't occur at all, or they'd occur too slowly to maintain life. So enzymes are needed in order to maintain life. Enzymes are also considered catalysts. So if you do chemistry, you know that a catalyst is any substance that alters the rate of a chemical reaction, but doesn't alter the products that are formed or being used in the reaction. Okay, so enzymes, type of catalyst. But what makes up an enzyme? So enzymes are made up of proteins, and each cell makes its own enzymes. So whatever enzymes are needed in that cell will be made inside that cell through protein synthesis. Every enzyme that exists is specific to a particular reaction. Okay, so for example, lipase is an enzyme that is specific to breaking down lipids. All lipases will have that specific structure that only lipase has. Okay, this means that if we have a metabolic reaction occurring in multiple different steps, which is how they work, each step will be catalyzed by a different enzyme, because each step will have different products and different reactions. Whatever type of enzymes the cell produce will determine what type of chemical reactions are going to happen inside of that cell. There's 2 different types of enzymes. There's enzymes that will catalyze reactions inside the cell, and we call them intracellular enzymes. And there's enzymes that will speed up reactions outside of the cell, which we call extracellular enzymes. How exactly does an enzyme become specific to that reaction? Well, as we know from last week, proteins are made up of amino acids. And we've got here, those amino acids join together to form a peptide, and then multiple peptides form together to form a protein. So we're looking at the quaternary structure here of the protein. Okay. Whatever that sequence of amino acids in that protein will cause that protein to have a particular 3D shape. And it's that 3D shape that gives the enzyme its specific function. Okay, so that whatever amino acids make up that protein that makes up that enzyme, that's determined what that enzyme is going to do in the cell. How enzymes work. So for those of you that don't do chemistry, you may not know how a chemical reaction occurs. You always start with reactants, and you always finish with products. So these are the 2 things that are going to react together to create this. Okay, for molecules A and B to actually bond together and react together, they have to collide in a certain way that they'll fit together and the reaction will take place. That's the only way we can get C and D. So without catalysts or without enzymes, we have to wait for them to, by chance, collide into each other and bond and react. Enzymes increase the chance of that occurring because they force the molecules to sit together in that one spot. They might put strains on the bonds, which means it's easier to break those bonds, or they could just make the reaction happen in smaller steps. Okay, but enzymes' job is to make it easier for these 2 to collide and react. That's how an enzyme works. A little bit more on how enzymes work. So we talked about different types of proteins last week. Enzymes are globular proteins, and they have a region on their surface, this region here, that we call the active site. Okay. Now this shape here is complementary, like a jigsaw piece, to the reactant or the reactants that the enzyme is going to catalyze. Okay. We call the reactants when we're talking about enzymes, we call them substrates. So this here is a substrate, and you can see it complementary fits into the active site of this enzyme. That's how we know this enzyme will act on this substrate, because they fit together like a jigsaw puzzle. Okay. Now, if you ever asked in a test or exam how do you define what an active site is, we have a definition down here that's really good, and I would encourage you to memorize. So the active site is the region on the surface of an enzyme molecule with a particular shape into which only the substrate molecule with a complementary shape will fit. Okay. Put simply, if a substrate doesn't fit into the enzyme, that enzyme's going to do nothing for the reaction. You have to have the right enzyme for the right reaction. That's why specificity is so important when we're talking about proteins. Now, truthfully, if we go back for a second, it's not just a nice, easy fit here. Like this is a perfect fit in this picture, but that's not really how it works in real life. What actually happens is you have something called the induced fit molecule, where you can see the enzyme here has the complementary shape to the substrate, but it's not perfect. Okay, so in order for the substrate to bind to the enzyme and form something known as the enzyme substrate complex, the active site needs to change shape slightly to give it a better fit. That's why we call it an induced fit. It's a forced fit. Okay, so the substrate will fit into the enzyme here. The enzyme will slightly change shape to give it that perfect fit in order to form the enzyme substrate complex. Once the reaction takes place, the molecule will then break away. The enzyme will remove itself from that induced fit, and it can be recycled to work on another substrate. So enzymes can be recycled. Okay. That doesn't mean that enzymes can't be destroyed. Like proteins, because they are a type of proteins, they're very sensitive to the change in their environment. So they can be altered by temperature, how acidic it is in the environment, so the pH level, as well as if there's a chemical inhibitor, which we'll talk about in the next few slides. Okay. Each different enzyme is going to work best at different temperatures and pH levels based on where they've been designed to fit. For example, any enzymes that are created inside your stomach are going to need to be able to operate in quite acidic environments, whereas enzymes that are inside your intestines, for example, they're not going to want that acidic environment. They're going to want a slightly neutral to alkaline environment in order to work best. Okay, so every enzyme will have its own optimal conditions. Any changes to those optimal conditions will cause the enzyme to change shape, which we call in biology, the enzyme is denatured. Okay, if the shape of an enzyme is altered, or if the enzyme is denatured, that enzyme can no longer function as required and needs to be replaced. So things like increasing the temperature too high, which is what happens with some illnesses. Our body rises our body temperature to fight off the infection. We raise our temperature too high, it starts to denature the enzymes, and all of a sudden the enzymes can no longer function. Your chemical reactions don't take place as fast as they should. That's why your body can't increase its temperature too high. We talked a little bit before about inhibitors. So there's two types of inhibitors. They're competitive inhibitors and there's non-competitive inhibitors. Okay, so first off, what is an inhibitor? It's a substance that has a very similar molecular shape to the substrate, and it can actually block the substrate from entering an enzyme and make the enzyme ineffective and not able to do its job. So competitive inhibitors, we've got over here, and I apologize, the video is blocking this a little bit, but competitive inhibitors will actually block the active site, and we can see that here. So here we've got a competitive inhibitor, and it's blocking the active site. That means that the substrate can't physically get inside this enzyme, and it has to go find another one. We've got one less enzyme in the body that can perform that function. Okay, whereas a non-competitive inhibitor doesn't bond to the active site. It's not competing for that active site. Instead, there's a section called the allosteric site down here at the bottom of the enzyme, or it could be on the side of the enzyme. And the non-competitive inhibitor bonds in there. Now, when it bonds in there, it changes the shape of the active site. The actual protein itself changes. And now that this enzyme has a different active site, the substrate can no longer fit. So it hasn't blocked up the active site, it's physically changed it. So competitive inhibitors block the active site, non-competitive inhibitors change the shape so the substrate can't fit. Very slight different topic here. We're going to talk about exogenic and endogenic reactions. So we know that all chemicals contain stored energy. And the reactions, when reactions take place, like when enzymes help them take place, it changes the amount of stored energy in those molecules or in those chemicals. So an exogenic reaction is a reaction that's going to release energy. So the reactants are going to have more energy than the products. Okay, so we've got more energy to start with than we did at the end, because energy has been released into the environment. An endogenic reaction is one that's going to consume energy. So the products actually have more energy than we started with in the reactant. It's absorbed energy from the environment. Okay, two examples of those. So glycolysis is an example of exogenic reaction. Now we haven't covered glycolysis yet, but we've got the definition down here. So 2 molecules of pyruvic acid are produced for each molecule of glucose broken down. Glucose has more energy than the 2 pyruvic acid molecules, therefore releases energy, and that energy is used to make ATP and provide our body with energy. So glycolysis is part of aerobic respiration, providing energy for the body, and that's because we're breaking down glucose, our products have less energy, energy is released into the environment where we can use it. An example of an endogenic reaction, on the other hand, is protein synthesis. Amino acids are joined together to form long polypeptide chains, but that requires energy. So we break down ATP and we take the energy from the environment and put it into those polypeptide chains. The products have more energy than the reactants at the beginning. And lastly, we need to talk about activation energy. Okay, so this is basically all reactions have something called activation energy, and that's how much energy we need to start the reaction in the cell. Okay, enzymes speed up chemical reactions by lowering the activation energy, and as we said before, they can do this by bringing the reactants together, so there doesn't need to be as many collisions. They're literally just holding the 2 chemical or 2 reactants in the right place so that they can react and break or react and bond. They might bind to a substrate that puts strain on the chemical bonds, meaning the substrate is more likely to react because they want to break those bonds. There's a lot of energy on that bonds forcing it to break. Or they could make the reaction happen in smaller steps, and each step requires less energy than the overall reaction needs. Generally, in a reaction, the steps are reversible. If that's the case, the same enzyme will catalyze both the forward and the reverse reaction. So it can either bond the reactants together to form the product, or it can break the product or break the reactants to get up to form the products. Okay, it can either build up or break down. Whatever the concentration is of the reactants and products in the cell will determine the reaction that will occur with that enzyme. So if you've got more products, then it's going to work breaking them down. If it's got more reactants, it's going to build them up, for example.